Hello friends, in this video I'll be talking about gluconeogenesis, okay, the Malate shuttle in gluconeogenesis. Okay, so before going on to our Malate shuttle, I'll first like to talk about gluconeogenesis. Okay, so this process of gluconeogenesis is the process in which the non-carbohydrate source are utilized for the formation of the carbohydrate compound. Okay, and those non-carbohydrate source are the lactate, okay, the glucogenic amino acid, which is alanine, okay, and then we have glycerol. Which is formed from the breakdown of triacyl glycerols by hormone sensitive lipase. So these triacyl glycerols consist of the free fatty acid chain and a glycerol molecule. So the free fatty acid, they are utilized for the beta oxidation purpose, and the glycerol is used in gluconeogenesis. So 95% of the energy comes from beta oxidation of fatty acids, and glycerol contribute to 5% of energy. Okay, so this gluconeogenesis, the process of gluconeogenesis is reversal of glycolysis. Only some of the enzymes in gluconeogenesis is different from glycolysis. Okay, and the name of those enzymes, they are pyruvate carboxylase. Okay, then we have phosphoenol pyruvate carboxykinase. Next we have fructose 1,6 bisphosphatase and glucose 6 phosphatase. So these enzymes, they are different from glycolysis. Okay, and the last step of glycolysis is conversion of phosphoenol pyruvate into pyruvate, which is an example of substrate level phosphorylation. Okay, but during gluconeogenesis, the pyruvate cannot be converted into phosphoenol pyruvate by the same enzyme. Okay, because it is a irreversible step in glycolysis. So pyruvate to phosphoenol pyruvate conversion is facilitated by Malate shuttle, and the process of gluconeogenesis is interdepartmental. If I say interdepartmental, it means one or two steps it happens in mitochondria, and rest of the step it happens in cytosol. Okay, so we'll be discussing about the Malate shuttle in gluconeogenesis, how the Malate shuttle convert, or how the process of Malate shuttle facilitate the conversion of pyruvate into phosphoenol pyruvate. Okay, so the process of Malate shuttle, it happens in mitochondria. Okay, it happens in mitochondria. As I told you previously, there are multiple sources of non-carbohydrate source that are a part of gluconeogenesis. One of such is the amino acid, which is alanine. Okay, and this alanine is converted into pyruvate by the process called as transamination. Okay, and the process of transamination require pyridioxyl phosphate as a coenzyme. Okay, so in Malate shuttle, if the alanine is a precursor for the conversion it into a pyruvate, what happens is this pyruvate, they enter into mitochondria. The pyruvate enter into mitochondria. Okay, and first the pyruvate is converted into oxaloacetate. Okay, first the pyruvate is converted into oxaloacetate, and the enzyme for the conversion of pyruvate to oxaloacetate is pyruvate carboxylase, which require biotin vitamin as a coenzyme. Okay, Next, this oxaloacetate, it is impermeable to mitochondrial membrane. It is impermeable to mitochondrial membrane. So, this oxaloacetate is first converted into malate, okay, by the enzyme mitochondrial malate dehydrogenase. And in this process, the NADH is converted or oxidized into NAD plus. Okay, so what happens is, first the pyruvate is converted into oxaloacetate by the enzyme pyruvate carboxylase, and this oxaloacetate, which is impermeable to mitochondrial membrane, is converted into malate by the mitochondrial malate dehydrogenase enzyme, okay, which uses NADH as a coenzyme. After that, these malate, they are exported out of the mitochondria into the cytosol, okay, and this malate is again converted into oxaloacetate, So here again this NAD plus is reduced to NADH. So this NADH is utilized during gluconeogenesis, the conversion of 1,3-bisphosphoglycerate into glyceraldehyde 3-phosphate. Okay, right? Next, if lactate is a source, something different happens. Okay, if lactate is the source, something different happens. So this lactate, okay, the lactate, they are produced from the anaerobic respiration, that means when the body is exercising vigorously, and also RBC, which lacks the mitochondria, and therefore there it can't be undergoing aerobic respiration. So the when lactate is a source of gluconeogenic compound, or lactate is a gluconeogenic compound, that lactate is first converted into pyruvate by the enzyme lactate dehydrogenase, which uses, where the NAD plus is reduced to NADH in the cytosol. Okay, so this NADH will serve during the conversion of 1,3-bisphosphoglycerate into glyceraldehyde 3-phosphate. So there is no export of NAD plus out of the mitochondrial membrane, or NADH from outside of the mitochondrial membrane. Okay, so this NADH can be utilized during the gluconeogenesis. Okay, and when lactate is a source, what happens is the pyruvate, it enter into mitochondria. And this pyruvate is converted into oxaloacetate by the enzyme pyruvate carboxylase, which require biotin as a coenzyme. Okay, and this oxaloacetate is converted into phosphoenol pyruvate by the enzyme mitochondrial phosphoenol pyruvate carboxykinase. Okay, and this PEP carboxykinase, and this phosphoenol pyruvate is exported out of the mitochondrial membrane, and it can be further converted into glucose molecule by the process of gluconeogenesis. Okay, this is all about the malate shuttle. If you like the video, kindly like and subscribe. Thank you.